All right. Well, welcome everyone. Let's wait for another 2 minutes before we start. While we are here, maybe like we can share a little bit about our course right here. So for today is an optional class that we want to provide it so that we know what we are trying to do, and we also know what is our goal and objectives of um, the overall attempt of trying to work on this class. Um, Hi, good evening, Tanvi. So, so we see that like we have like a lot of people apply for this course from different countries in Africa, and also like a lot of countries outside of Africa. So myself, my name is Ice. I'm not from Africa. I'm from Thailand. But Kato and I get together and like discuss a lot about like how do we make this um how do we make the class that is accessible to everyone. So that's like how the SynBio4ALL Africa has started, because we want to make sure that the synthetic biology education is available for everyone. So with that, we have like a few of the past courses, and we're gonna tell you about that during this presentation as well. So just to keep in mind that we are trying to make this the most appropriate for the African students or course takers wherever you are, whichever stages you are. So if you're not from Africa, please you're welcome to stay with us. And but we are sorry about the time zone or anything that might not be perfect for you, because we are trying our best to like have the focus on the target group that we have. Um, but hopefully, like you can interact and we can engage and work together across the class. Right. I think we have a good number of people joining. Kato, you wanna share anything first before I take it to the presentation?
Okay, thank you so much, Ice. I am Sebunya Kato, and I would like to welcome all of you who have joined us in today's optional class. But this is like the icebreaker for our big course, the Plant Synthetic Biology course. And I hope you will like it, and whatever we shall share today, it will be rotating around synthetic biology in general. And please, if you have more questions regarding synthetic biology in general, please reach out to us, as ICE will be sharing to you, and we shall be happy to help you. I wish you good luck and enjoy the session. Thank you so much.
All right, thank you, Kato. So I'm gonna share my screen soon. And just for this session will be recorded, and all of our session will be recorded. We will keep like Kato and I will keep our video on. All of you, like if you want to keep your video on your call, you don't have to keep it on if it's gonna make your connection having a problem, because I know that like sometimes like having the video on make the connection go harder. So I think that's fine that just a few of us keep it on. With that, let me share my screen real quick. Let's see. Kato, can you help confirm if you see my screen? You
Yeah,
muted, but I think you said yes, right?
I see your screen. Yeah.
All right. Well, thanks a lot, everyone. So again, I want to introduce myself. My name is Ice. My name is Cholpisit. Full name is Cholpisit Kiattisewee. Is not that easy to pronounce, but I have the same problem when I'm trying to pronounce my African friend's name as well. So I think we share the same trouble together. So I'm a volunteer for this with the SynBio4ALL Africa. I think I started volunteering since 2022, 2023, right, Kato? We get together and try to plan for this course. Maybe it took almost a year for us to figure it out what we should do. And then we were like, it's not going to be perfect. Let's just try to do something. And that comes to, that the course that we were provided in 2023. That was 2 years ago. So with that, we sort of like learn to a certain extent how to make sure that we can make the class that would be appropriate for all of you, and also like help us as like the volunteers learn together as well. So a little bit of background. I'm originally from Thailand. I did undergrad in Thailand. So I work in the space that was like, we were pretty close with nature, and I like plants a lot. I like eating them. I like to, get into the wild and see a lot of things. That's why I really appreciate plants, but I'm not a good plant breeder or plant engineer. That's why I'm in this class trying to help coordinate and trying to help with the other simple and synbio things. I come to the US, to Seattle, at the University of Washington to work on my PhD, and I graduated just last 2 years ago, around 1 or 2 years ago. Now I'm a postdoc at MIT working on synthetic biology. So I work mostly with microbes, but my interest in plants remain, but it's not my expertise. So again, I'm gonna share about these like general aspect as much as I could, and then when the class actually started from next week to and on and on. We have more experts who are studying on plants who can share with you more about the specific details. So today's class is optional because it's just a course orientation. So if you miss anything, it will be recorded and it's going to be shared with all of you. So if you have to go to move to other tasks, please feel free to. One kind of key things that we are going to try is that I'm trying to go through the past courses and trying to provide some recap of what we have done in the past 2 courses. So there are a lot of them, and I wish that all of you have time to go through all those recordings. But if not, I wish that like this kind of like 15-minute session could be good for a kind of recap and to introduce, and maybe if you're interested in some part, you can go back and take a look at them. So that's why we call this week 0. So I forgot that I want to show this kind of like schedule. So we plan this session for just 45 minutes, but it may extend. If you have any question, just feel free to raise your hand or like type into the chat and we'll work on it. So just keep in mind that if I don't see or I don't respond, feel free to just unmute yourself and interrupt me and I'll just stop. All right. So we're gonna have like around 20 minutes to talk about the background of the SynBio4ALL Africa and some of the course objective that we're gonna do for this year. We're gonna take a few minutes to navigate some of our available resources from the past years. And we'll be reviewing some of the synthetic biology and something about plants. It's gonna be very quick because we wanted to spend more time in the actual class and just like give you some earlier ideas. So we're not going to go too far about plants yet, but we want to make sure that you know why we need to do plants and why we want to study plants. So, all right. So there's a logo. Is a new one. And FAI, our website right now might be a little bit of like, in a novice, like beginners level, but we have like our volunteers coming in to help us to make our website look much better. So you're going to see a new kind of website soon. So with that, this is our link to the website. If you search SynBio4ALL on Google, they should show up. Maybe like if you add SynBio Africa, it should also show up as well. And one thing that we just added in is that we want to make a mailing list so that when we share any information or activities about the program, you can receive this information without us trying to just like go to social media. We want to send you a direct email instead. So if you're interested to join our mailing list, you can click join us. If you can see my cursor is the join us button right there. And I'll tell you how to join technically. And the other thing that we are trying to share is that like we have several activities going on with SynBio4ALL Africa. So we have the course, of course, like we have like, the beginner course in 2023. We have the microbial course in 2024. We also plan to have like a lot of the webinar going on every month. We had some issue in the past few months that we were so busy we cannot get some time together with our volunteers. So we will just bring it back again. Started from last month and also like the next week. I'm gonna talk about that as well. We also go to conferences. In this case, I volunteer to attend the EBSC conference, where a lot of us were partnered with EBRC, Engineering Biology Research Consortium. And for example, myself is working with EBRC a lot as a student and postdoc organization. And a lot of our volunteers that are going to be teaching you in this class is also volunteers from the EBRC itself. So that's another global organization that we partner with. And another reason why we may want to get into the Plant SynBio right now is that in last year, there are a lot of interest in the Plant SynBio. So for example, if you look at some of the final projects that were posted on our website, so we did some final project. I'm going to talk about that later, what it is. But a lot of those things were mentioning something that was relevant to plants. So these are just some that were shown, but we actually have a lot more projects that we work together. So that means like if you do something about plant, it's going to be a pretty good interest for everyone. That's why we sort of like think about what should we do instead. We were thinking about maybe computational and also plant, but we decided to move to the plant one because we see an opportunity to work with the Experiment Foundation to get some support. So this is like the micro grant that we submitted. We worked together to submit to the Experiment Foundation so that we get some support. And maybe we can do some of the in-person kind of like lab work together. So if you're interested, I'm going to share this slide so like you can get into this link and read further what we are trying to do. And I'm also going to explain that briefly. So the things that we committed is that we want to make this course open source, which is something we have been doing already. So that's not too hard. It's going to be check mark already. The second thing is that we're going to identify the plant for the whole genome sequencing. We want to make sure that we can identify some plants that is going to tell us something about the biodiversity in Africa. And this kind of like platform could be applied to other places as well, we believe. And then we want to initiate some of the lab work locally in Africa to secure some of the plant materials, and then get the DNA extract and then send them for sequencing. And then we're going to figure out like what can we learn from those kind of sequencing. We are trying to go for the plant that is not known before in terms of genome. So we're going to be partnering with a lot of organizations to make sure that we are in charge of those things. All right. So that's some of the, the video that I made. If you want to hear, another recording of my voice, that is going to be available right there, but I'm not gonna go too far into that one. Oh, I think people were asking maybe like if we can share the link. Let me see if I can click on, and please let me know if you can see the link right here. Were you able to see? Kato, can you help me confirm if you can see the web page?
Yes, the web page is visible.
Yeah. So if you click on that link, you will see this web page that narrate like what is the project and what we are trying to do, and also like it's gonna list some of the plans that we have. As the class goes, we're gonna go and update some of these plans as well. All right. So I think that's it. Oh, okay. Thank you, Innocent. I think like some of the letters is too small to see. So like some of them is going to be a screen captured. So if you cannot read, maybe you can take the slide and take a look further. But I'm going to talk about most of the things that that were mentioned here. So please, bear with that. And we will share these slides so that you can see it later. Right. Okay, so this is our instructor team. We are a team of like more than 7 or 9 people volunteering with us. Right now, we have a lot of people taking care of these 10 to 11 classes. So this is week 0, and we're going to have week 1 to week 10. While there is 1 week that's going to be dedicated to the group project, we're going to use that week to evaluate what we need to learn, what we have learned, to make sure that we can identify the plant that we're going to work on the open source genome sequencing data. So these are the people that volunteer with us. You see them across the weeks. We also have these syllabus. So we want to make sure that you see all these syllabus as you work with the class. If you are looking for any information, you cannot find it, it might be in the syllabus. So that's something that we want to make sure that you are aware. And another disclaimer that we may have more volunteers coming in, we also want to make sure that all the accessible or the education that we're trying to do is accessible. So it was designed for the low resource community. If you see something that might be able to improve, please let us know. We want to make sure that you can access them as easy as possible. And we believe that like people from outside of Africa are welcome to join as well, as long as they can access the system with their available resource. So in extension to what we are trying to teach in the class, we also invite a few professors who are working in the plant synthetic biology to come and share with us what they're working on. So next week, 2 days after we finish or wrap up our class, Professor Katalin Monishak will come and share with us about the plant SynBio and some automation work that Professor Katalin is working on. So the other thing that Professor Katalin is working on a lot is cell wall bioengineering. So you see some presentation and some discussion of like how to get further into the plant cell wall. And that's something that was pretty unique to plant compared to other microbes. And we're going to touch on that a little bit. So again, I want to make sure that I show you how to join the community. If you want to join the community, what you can do is go to that first page that I just showed. Maybe like I'm going to jump to that one again. Let's see if I go to the right one. Right. You see this join us button. If you see the join us button, then it will bring you back to the page that you're going to see next. Yeah, it will bring you to this page. And if you go to this page and you click onto this post right here, this like joining SynBio4ALL Africa mailing list, it's going to show you how to join. And it's actually like what I just showed you here, that you should be, clicking join group right here. It's going to show the button, or like, whether you want to receive all the email or you want to receive some of the emails, choose however you want, and then you'll be part of our community. So whenever we have the webinar, we're going to share that with you. Again, by participating in this course, we are using the Google slide. Oh, sorry. I think something is weird on my end. It doesn't want to stay on the other slide. I'm sorry for that, but like that slide is the one that I'm trying to show you. Maybe I'll just use this trick. I hope you can see this. So I'm showing you this slide from my PowerPoint instead. I have something wrong with the slide. So all of you probably have received an email to join this course. If you didn't receive it, it might mean that your email that you provided in the registration is not compatible. So if it's not compatible, please like reach out to us or just like apply for the course again. We still keep it open, and make sure that you use the email that is compatible with the Google. Maybe the base is to use the Gmail account. And then like, we will email you the Zoom links and reminder maybe 3 days or 1 day before the scheduled class, or also the webinar that might be happening. All right. With that, the goal of participating in this class is to make sure that you can look through and learn from our lectures. We are not taking participants from the lecture, so you're free to join or watch the recordings at your available time. So with that, the class will be provided at this time. So it is 4 p.m. UTC, 7 p.m. East Africa time. If you think in terms of the US time, it's going to be 9 a.m. in the Pacific, in the West Coast, and 12 at the East Coast where I am right now. So the Google link invitation should have all the information, and we'll keep sending you the invitation for the whole event again in the email. So make sure that you're part of the Google course and you get the information. And also check at the syllabus for any information that you might be missing. We share all the slide presentations, all the recordings, and the homework for each of the class. So we also want to provide a certificate of course completion. It's unfortunate that not all of the participants so far will be able to complete everything. That's why we want to make sure that like for those who can complete everything can get a certificate for the course completion. So with that, what you need to do is that you take the lectures, which is like we're not counting. So it's your call on how you want to take it. You can, do it later in the course. That's fine. But we want to make sure that everyone work on the homework. The homework is not going to be difficult. It's just going to be a recap of the question of like what is going on in the previous class. So in the syllabus, there'll be some more explanation about the homework, but we want to grade the homework by the next class. So that means like if you take the class like regularly, then we would appreciate that you submit the homework before the start of the next class. You can submit it late, but it will not be graded until maybe week 7 or week 8. And we want that you have like around 5 points out of 15. And if you don't get 5 points, there is a chance to work on the makeup homework to make sure that you can pass. But it would be much better that you don't have to do it, right? So keep in mind that like there are some homeworks to work around. We have a pretty serious concern about using AI on the homework. So we believe that like you can use AI to or like the generative AI to help with your class or learning. Like we have to use them appropriately. We just ask you that please don't just copy and use that as your answer directly. Use it to learn and maybe like take what you learn from your discussion with AI and put that into the answer. We're going to provide that kind of like example in the example homework that we're going to put at the end of this class as well. And lastly, we want everyone to participate in the group project. So in this group project, it's similar to like what we are trying to work on, which is like the identifying the native plants for the biodiversity study. So more information will be described and explained through emails and other notes throughout the class. So we're going to work on this together to identify what plant would be interesting for us to work on. All right. I think that's most of the information I want to share for now. If you want to look into any deadlines, please look at the syllabus and they will have all the information provided for you. We'll send the email with the syllabus link again, just to make sure that everyone has access. All right. So let's go to some available resources that came from, our previous classes. So in 2023, we worked on the beginner course. So the intention of the beginner course that we work at a time is that we want to make some foundation. We want to make it very simple. So you see that like in this syllabus of the 2023 beginner course, the question is very simple. Like what is synthetic biology? What is central dogma? How to control what cells make? What is the useful parts in scenario? What is the laboratory techniques, and maybe like some kind of like far end like the biology by the numbers or intro to some CRISPR. So so in this case, if you haven't taken the beginner course, you are free to watch through all the recordings. Is provided on the website. So this is the list of the contributors to the introduction to the beginners class. Everything should be available right there, and you'll be able to take the slide, the recording, and also the homework. And then these are like some snapshots of what it is on the website. So there are 6 of them available. You can just watch through them on YouTube. And these are the slides and homework that were available right there. So please work on it at your own pace. Not you. I feel like if you walk through all of them, it's gonna help you a lot in terms of like learning other classes, because sometimes we want to go further into the lecture. It's actually a lot easier that we have some of the basics, but you don't have to take the beginner course to start the Plant SynBio, because we have some recap, including this class that we're gonna have some recap. So if you don't have time, please feel free to just go back to it only when you feel like you need to, right? So if you have some question, you're concerned like, I don't understand what is going on with this thing, can I go and learn somewhere? that's where we want to point you to, which is the beginner course, which we try to make it much more accessible and much more simpler than what we're going to walk through for the plan, because plan is not easy. So the other part is that like we shift a little bit further from the beginner to the microbial. So we are trying to work on the microbial synthetic biology last year, and we have 7 lectures plus some kind of group projects. So I want to emphasize that like, the lectures is gonna be a little bit more different. So we work on, a similar course review, and we focus on engineering of different parts. So we start from protein, this bacterial strain, and the metabolic network, and then we go for the application side on the frugal science, on the bacterial therapeutics, and also the sustainability approach of the microbe engineering. So these are all interesting talks. So if you're interested in, please give it a look. And if you're interested in some of the group projects, a lot of these group projects were also made available on our website. Some of them were highlighted, but actually more of them on our YouTubes. So you can scroll through. We asked all the students that like we want to make this available. So like, if you see some of your friends who are taking class right now who were presenting on some of these things in the microbial class, and you want to chat, you want to reach out more, please feel free to interact. If you're wondering how do you interact with each other, I think Kato will help organize the WhatsApp group, that which is like we found that it's the best platform for the African countries. So we will send more information about WhatsApp soon in the email so that you can join the WhatsApp community. So, like another 7 courses are available right here, and they were taught by different people. So sometimes some of the materials were overlapping, and you may find some of the teaching to be like interesting in the different approaches. So I hope you enjoy some of these recordings. All right. With that, I think we are pretty good on time. I'm going to move to some of the recap of the synthetic biology that you can find in all the lectures that we presented in the last 2 courses. But I want to make some recap on, what could be available, what could be fun here. All right. So again, just to make sure, if you have any question, please feel free to write onto the chat, and I'll just like try to answer as we go through. And also maybe like I'll just stop and jump to explain more what is going on with some of these parts. But for now, I think I'm gonna move through our review session right now. And hopefully like, in 15 minutes or 20 minutes, we're gonna be done with this, and we can come back and have more open discussion with everyone. All right, so for the central dogma, I think the central dogma is something that was like kind of like not a regular term. It's not something that people always think about even when you learn English. But the central dogma, the meaning of it, is actually the basic principle. So the basic principle of molecular biology is that like most of the time the information will be flowing from DNA to RNA and to protein, and then they will turn to some of the cellular functions. With that, we can consider like, if we work on our computer or a phone, we can consider them as like something that was stored in the device that was more like DNA. If we want to transfer it from one place to the other, they will be copied into the RNA. So that sounds like something that you're gonna give some information you have into your friends. And then if you turn these kind of information into function, it's gonna be a protein. So if you think about the application on your cell phone that was running the Zoom call right now, then that's kind of like how the protein will functioning inside the cells, starting from how they store information in the hard disk or some other thing. The basic concept of synthetic biology is that we're trying to control them. So let's say if any organism doesn't make the green fluorescent protein, but we want it to make it. So the green fluorescent protein is a protein that if you shine light onto it, it's going to emit the green light back. If you put it into plant like this is how it's going to occur. So what you're seeing here is the dark space, but you shine some blue light onto it. When you shine some light onto it, the part of the leaf that have the GFP is gonna return the green fluorescent back. So this is different from the greenness of the leaf, right? Because usually it's the color, not the fluorescent, not the color that was like glowing in the dark. So this is something special that you can use synthetic biology or biological engineering to work on. So this is an example, but like synthetic biology is more than that. It's more about how to predict, how to program, and how to control them properly. With that, if we take a step back and look at the big picture, this is a very nice picture from the publication that came out last year about like how do people usually study synthetic biology. So there are actually two ways that you can work on. You can deconstruct. You can work on something that you want as a big picture, and you just like make it smaller into a smaller part and see what you can make change in the smaller part to go to the upper changes. So this can be the environmental one into the health one. So the other approaches is to do the reconstruction. So you take some of the sequence, you take some of the information and just reconstruct them, combine them into a different system, or just build them from scratch. So in that case, you may start from the molecular level, and maybe like you're gonna form them into some sort of network, and then put them into the organism or into their DNA, and then make it work. So a simple example that was mentioned in our first classes is these two, which is one of the two molecules, one of the two products that are very important that was relevant to biological engineering, genetic engineering, synthetic biology, or metabolic engineering. These are always an important example of the field. So in the past, in maybe like 50 or 70 years ago, when people have diabetes, when your body cannot control the sugar level in your blood, what you are missing is actually the protein called insulin. So this insulin is a protein that our body just read from the DNA and make it into RNA and then protein. So this protein is functional to control the sugar level. If our body cannot make it, we have to find it somewhere. In the past, people just make it from pork liver. So if you see this pile of pork liver, it just turned into just a single tube, a very small tube of insulin. And that costs a lot, both in terms of money and also in terms of like the amount of animal that you have to, harvest them to get these compounds. It's not that sustainable. So when people study genetic engineering, they found that you can take the gene, the DNA of insulin, and put that into the bacteria. When you do that, what you have to do instead of like breeding or like growing a lot of animals, you can just grow the microbe, which grow a lot faster and use a lot less resources. And microbe doesn't have to torture that much compared to the animal. So that's a lot more humane. With that, you can just break down the microbe, break down the E. coli, and then you'll be able to get the insulin to use like that. If you take it a little bit further, if you want to make a compound, a small molecule, or something that was like medicine that could come from plants, for this one is artemisinin that was used as anti-malaria drugs. So for those of you who are living in the tropical countries, you should be aware of the malaria, right? It's kind of dangerous. So if you want to make more of this by extracting them from plants, it takes a lot of time. And sometimes if the weather is bad, then you don't have a lot of plant that year. So the price of the drug is going to increase a lot more. So if you can put these artemisinin production genes into some yeast that we use to brew the beer, then they can instead not just brew beer for us, but instead brew these medicines so that we can extract and then use them to treat our disease. So these are the two, I would say, like epic pinnacle examples of the synthetic biology and metabolic engineering that has been transforming how we live in the past like 50 or 20 years. So with those two examples, I want to come back again and want to emphasize that like most of these biological data are stored in the DNA. And with that, if you want to engineer anything, you may have to engineer at the DNA level, and then your engineering at the DNA level will get transferred into the RNA or the protein and function as they're supposed to. And most of the time, this DNA will be engineered in the form of plasmid DNA, which is something that we do the engineering in the microbes. If you go to plants, it could be a little bit different. But whatever processes we're going to do in the plant, we will have to do some initial engineering, mostly on the plasmid level. So in terms of the parts of the plasmid, it was actually organized from different parts. And if you want to see further on like, what is available out there in the plasmid, feel free to take a look at some other recording. But in brief, we can consider the DNA as the different parts that fuse together, and they will have different function. So in this case, I took it from the slide that focused on the Golden Gate assembly by fusing different parts together. So you see that like they have some annotation from this paper when they're trying to put different DNA together. There'll be some that work as a coding sequence. There'll be some that work as a promoter. There'll be some that work as a terminator. So the way that works is because like when the DNA has to be read to turn into RNA, they need to identify where the promoter is. And when the promoter was identified, it's going to read the DNA sequence downstream, and it's going to read the whole thing until it finds the terminator. And this chunk of like number 2 to number 4 will turn into the RNA. And then the RNA will be translated into protein, so that can make the function later. So this is an example, an overview. It may not be extremely like precise or correct in terms of like where it is. And if you wonder where the exact part is, please feel free to take a look at some of the other recordings about the DNA parts. I think is on our beginner class on the useful parts of SynBio. Please feel free to take a look at that. The other approach of engineering is that we can also do some precise engineering at the DNA level without going to the plasmid. So one good example is the CRISPR process that received the Nobel Prize in 2020. It was known as being the molecular scissor. So what it can do is that like if you think about having these two system of DNA available, if you try to just keep breaking them, at some point they can make some mistake and lose some of the part, and then they're going to lose some of the function as well. And on the other hand, if it gives them some of the template to repair, let's assume that like you keep breaking the strand of the wood, for example, and then you put another wood back on. Sometimes it's going to be able to like connect a new branch onto the existing branch, and that's how it works. So these are some kind of like the CRISPR system that has been going on. I'm not going to go into details. If you're interested, please take a look at maybe like course beginner course lecture number 6, and we have some more explanation right there. Something that we can also turn it into some kind of like clear explanation is that we can change the gene of color of the flower. So for example, if you think about the bicycle, if you want to stop it, you need the brake, right? If you get rid of the brake, then the bicycle cannot stop anymore. And that's similar to like how the color of the gene for the color of this flower, if it's expressing the gene that turned it into the purple or the blue color, if it was disrupted, it was deleted, then it's going to turn into something that was more on the white side. On the other hand, we can sort of like change how the brake works from the front side to the back side or vice versa. So in this case, you can maybe change the way the flower gene and the leaf color, the gene for the leaf color work, and try to switch them. In this case, you can see that like if you take the plant that has the flower that was blue or purple in color, but you take some of the gene of that turns that gives some green color of the leaf into the flower position instead, then it may change the color of the flower from blue to green. So that's some kind of like engineering that you can do with the precise genome engineering. I'm not going to go too far because like what we want to clarify again is that like all these engineering usually occur at the DNA level. And we have to be aware that the organism inside our planet is kind of diverse, like similar to like as human that we live everywhere. We want to make sure that we know what is going on in the different community of the organism or the different system. So in this case, we have some microbes, we have some bacteria or yeast that we're doing different thing. If we go to plant, it could be different as well. Or if you go to like a much bigger phyla, like a much bigger system, like animal or like insects or mushroom, they're also different. So with that, you have to know better on like, what is the most appropriate way of engineering these things. So that's why we care about this class. The other thing that we want to emphasize in terms of like the difference of the bacteria that we used to teach in the last course to the plant cells that we have here is that there are key difference in terms of the size of the cells and the size of the genome. So in this case, so we'll make if we compare, these bacteria and yeast together, you see that bacteria and yeast were around like maybe like 2 times or 5 times different in terms of size. If we compare these microbe size with the big yeast, with the big animal cells, they're around this size. And the animal cells is like around the same size as the plant cell, but they're in a big range. So as you can see here that, some of the cells can be visible to the human eyes. Like if you see some of the eggs, like let's say frog eggs, human eggs, chicken eggs, those are mostly just a single cell and they're visible to us. But you take a look as a smaller portion, the way that plant kind of breed or like communicate with each other, they sometimes use the pollen. And you see the pollen that were flowing around or like where pollen that came from the flower of the plants, those are also like sometimes a single cells. So that means like, the cell can be like a very big in size or something very small that you can't see from your bare eyes, like the bacteria or the virus. So these are, a major difference of the microbes and the higher organisms like animal and plants because they are so big. All right. So the other thing that we have to be aware of is that plant, in terms of like the genome size, is much bigger than other things. So something that I want to maybe like highlight right here is that, these are the microbe, like bacteria genome. They're pretty small. The number right here is 10 to the power of 6, around like 1 million base. If you're looking at something like fungi or mold like mushroom, you see that the size of the genome is like at least tenfold, like 10 times bigger. So that means like it's going to be relatively more difficult for us to sequence or to read and figure out what is going on with them. If we keep going further, you see some animals right here. Worms are not that complicated. Some shrimps or some sea insect are not that complicated. Insect is a broad group. They have different size of genome into different order of magnitudes. For mammals, they're around this size. And for plants, it's so broad. So it's not just that they can be very big. Some of them can be very different in size. Some of them may have like a lot of number of chromosome as well. So that's one of the reasons that if we were to study plant, we have to be focused. We have to make sure that we understand the context. We have to be aware that they may not be the same as what we are trying to study on the microbe ever. So this is like, a simple point I want to raise in terms of like, why do we need to care so much about plant? Why don't we just study cell biology as a term and just like ignore and just like keep using the learning to anything? It's because they're so different. That's why we have to beware of the context. We have to keep in mind that if we can do it in the bacteria, it may not be always as easy to move into the plant or vice versa. Something that works in a plant may not work as great in the microbe as well. That's why making artemisinin from the plant genes into the microbe is not that easy. It takes like almost 10 years for the scientists to complete that. All right. The other thing I want to mention is that like, if you take multiple microbes or multiple organisms, we can make some kind of biome or microbiome. So there are some different microbiome that exist in human body. There are a lot of microbiome available. In the ocean, there are some microbiome. In this class, we probably will be touching on some kind of soil microbiome where the plant is living in. So in most cases, a lot of interaction with the microbes inside the soil will interact with the plants by sharing some resources, by inducing some kind of changes in the system. So there will be more discussion on the microbiome. I want to bring it into this point first. We haven't covered too much of the microbiome part in our previous lecture, but we will be touching on the microbiome part in one of the lecture in our plant course here. And the other thing that I want to emphasize from what we have shared in the past course is that we want to make sure that our course are openly available. So this is the slide that I took from our friends who taught in the previous class. So these are open enzyme collection. So it's part of the collection that was a global collaboration by the Reclone organization as well. So Kato and I have been talking with some of the people who came from Reclone, and we have a pretty good relationship with them. So in this case, it is possible to share some of the resources if you move into the further system that you want to do some of the in-person experiment, or like make some simple experiment or instrument in your system. So we can talk more about that. Getting to the frugal side, if you're interested in more of these discussion, you can take a look at the frugal science course. I think Prince explained it very well on what is going on, and I think Prince is based in Ghana, right, Kato? I think he's based in Ghana. So, like Prince can share a lot, especially if you're in Ghana, maybe he can share more resources to you as well. All right. This is probably one of the last slide I want to share. This is from Enrico, one of our volunteer last year. Enrico has been working a lot on trying to make microbe for the bioproduction. So this is one of the example from the microbial class that we have been focusing on a lot. We are trying to make sure that the feedstock that we can use to make the new bioproduction is much more diverse. So in this case, there some discussion that was like moving further away. If you start from sugarcane, you can get some glucose and you can put into some of the fermentation to make different products. While if you don't take just sugarcane, let's say you take some lignocellulosic biomass, which is like some other branches that was on the floor, before they get degraded by some other fungi, let's take them to do some processing, and then take these to let the microbe culture them as well. These might be able to make the price of the product be a lot cheaper, because you can start from not just the sugarcane that you have to extract glucose from. You can technically take any branch that was on the floor to incubate them and then make them into a value-added product. So this is a very important process, that like how do you make sure that you can use some of the thermophiles or some different organism that can make sure that they can take the substrate and move further. So that's the end of the recap that I want to touch on. So again, I want to emphasize that like our course will be focusing on like maybe 3 key things. We want to work on the lectures. Oops, sorry. We want to work on the lecture, and with this lecture, we're gonna discuss on like what native plant we're gonna work on, and we're gonna move on the whole genome sequencing of that plant at the end of the class. Maybe we'll continue the project for the next year, and we might be able to analyze the data together. So if you're interested, please stay with us to work on it. And yeah, I think that's the end of the class. This is some recap of like what I share of like how the class would be. There will be an optional homework for this class that I just like talk about as well. I'm gonna make like a few questions for you, just so that you know how to use the form, how to submit these things, and we can work together. I will share that by the end of the week. There's a lot going on in the US system right now that make my schedule a little bit difficult to organize. So I apologize. I plan to share it right away, but I'm not quite done with that yet. So I'll finalize that and make sure that I can share it by the end of the week. So hopefully like these are interesting for you guys, and please like join us in the actual class next week, and also in the webinar next week. Right now, I feel like if there's any question that I can clarify for the course, I think we can use that time for our discussion. All right. I'm gonna go look at the chat right now. Oh, but Bakunda, you raised your hand. Please feel free to unmute yourself.
Yes, thank you very much, Mr. Ice. That was a very good presentation, and we look forward to going through everything with you. I was just asking, you had actually talked about something about the optional on 2024, something about some course in 2024. I actually didn't get you there. I was requesting if you could just make a quick recap on that part. Yeah, that's it.
Can you elaborate that again? Can you say that again? You want me to share that, or you want me to explain how to go through that?
Yes, you had shared 2023, there was an beginner course, and you told us you can actually access the videos and go through. Then 2024, there's something you said. I didn't quite get what you said. Maybe it was my network. I actually didn't get anything from that part. Yeah, thank you very much.
I see. So technically, what was available in the 2023 class is also the same for the 2024. Let me go back to that slide. Let's see. So this is what we have in our 2023 beginners course, and we have 6 lectures right here. So you can take a look at the syllabus to be shared on the website. You can look through the recording, the slides, and the homework and work on it. I think I make all the homework to give you the response right away, so you can see whether it's correct or not correct. Maybe the system will mark it as incorrect if the spelling is wrong, but you can take a look at it, and if it's correct, then you sort of like, give you response to yourself. The same goes for the 2024, but we have 7 courses and also some other presentation from the students. So please feel free to look through these 7 weeks of lectures. There are some homeworks, recording, and other presentation available for you to take a look as well. But beyond that, we have these like the proposal-based final project that we worked together with the course takers last year as well. So that's another interesting adds-on. So maybe like just to clarify from your question, Mukunda, we actually have everything that 2020 course has, but we have other things, like we have additional things from the student presentation that we can share with everyone. I hope that helps.
Yeah, thank you very much. Maybe also one more small thing. If we are to do the homeworks in that 2024 course, okay, we know it won't like help, like you won't give us like, I don't know, can we get feedback on whether we have done it properly or?
Yeah. So I would say, let me try to get into the link right now. I think I can click on the link. Yes. The link actually like put me through this website right away. And then if we go into, I'm just gonna go into my class here. If I click on slide, it's gonna show me where the slide is and how it looks like right here. So you can look through the slide. You can also watch the recording right here. That was me presenting. And you can get into the homework part. And the homework part is gonna be these kind of like, it's gonna be organized into easy, medium, and difficult question. So when you submit these, you get the response right away, and that's like an automatic response that we provide. If you have more questions and you want to talk with us, please feel free to send us an email, and then we'll respond to your question accordingly. And if we have more time during the class, we can also touch on that. How does that sound? Yeah, hopefully, like, if you fill this information, if you answer this information, you'll get the response right away. So you can give it a try. I don't want to like, add the question right away, because it may give like a weird answer, but everyone can try it out. All right. So let's get to the other question that we may have. Let's see.
I have taken care of most of the questions in the chat. You can concentrate on the people who are raising up their hands.
Okay, good job, Kato. So if you guys have any other question, like if those of you guys in the chat still have other question and want to clarify, please raise your hand or unmute yourself to talk with us. Gladys, I see your hand raised. Please, if you have any question.
Good evening, San. Thank you so much for the orientation. I would like to know more about the group project. Can you please elaborate on how it's going to be?
Give me
I
one
think that's a
second.
question.
Yes. So for the in-person, we still haven't finalized it because we still have to identify what the plan is going to be. So the group project that we're going to work on throughout these 10-week classes is that we're going to give you information on what the native plant, we're going to give you information of like what the plant biology is. We're going to give you some context of how to engineer the plants. And at the same time, we include some of the plant diversity. That would be one of the important thing that we should be focusing on in this class because the plant is so diverse and we want to care a lot for the biodiversity. That's one of the things that we care for. There was also like a big concern for the African learners in the past few years as well. So with that, for the class, through just this class, this 10 weeks, we're going to work on identifying. We're going to work together. We're going to submit some form. We're going to have some discussion. And we will figure out maybe a few plants that will be good candidates that we can grab from. By the end of the class, we will have those plants finalized. And we will work together within our team and some other volunteers to see how do we work on the in-person work. How do we find a lab space and how do we find the space that we can acquire these samples and maybe work on the genome sequencing. That was something that we don't want to finalize yet because we don't know what kind of plant we're going to be choosing from. And that will be, affecting what is the final plant that we will be doing the genome sequencing as well. The tendency that we're going to work on is maybe like we might have to go to a few universities that we have connection with so that we can borrow some of the lab space to work on some of the like DNA extraction from plant or from the system and see if that can help like facilitate the work further, because we want to make sure that some of us can do the in-person work. So if you guys, the course taker, were not too far from university that we're going to do this in-person work, you are more than welcome to join the team to work on these plant genome extraction project. I hope that helps. It's kind of like a stepwise process. So we want to make sure that like we choose the right plant first, and then we're going to identify like the space to work on that. Like so far, we've been contacting a few labs in Uganda to work on this project, but we still have to get the final choice or like the candidates for the plant genome to work on it first until we can finalize where we want to work on. Yeah. Any other questions? I see some question in the chat. So the question on whether the recordings and the lecture are going to be on the website. So the lectures will be available for you who register for the course and will be available to everyone later at the end of the class. So we want to make sure that we go through the recording, we go through the slides and make sure that the homework were appropriate for someone who is going to be taking it without our guidance, without our help. So we have to do some works on that to make sure that it's going well before we put onto the website. That's why it's going to take a little bit of time for us to organize all the materials and make sure it's going to work fine before we share it with the global, with anyone in the community. So all of you who take the class, all of you who were in the Google group, all of you who have the access to the syllabus, will be able to see the slide presentation, we'll be able to see the recordings, and we'll be able to submit the homework within these 2 or 3 months. And at the end of the class, maybe like a few weeks after we wrap up the class, we will turn all these materials to be open source, to be accessible to everyone. But that would mean we're not going to be grading homework timely anymore. The homework is going to be more of the self-evaluate homework, if that makes sense.
Okay, another thing to add on that, if you don't attend in real time, like maybe if you don't move with the class in real time, then you cannot get a certificate at the end. But if you move with the class in real time, you can get the certificate. You may miss the class, but you may follow up the recording and submit the homeworks in time, and you'll be in position of getting a certificate. But when you miss that process, you will not be in position of getting a certificate.
Yep. So again, we have some backup. So the homework is due before the class of the class at the week after. But let's say you are busy for 2 weeks and you cannot make it, you can submit it within the grading time that we set. So I think we have the time pointed out on week 7. I can't remember the exact date, but at the end of week 7, we will not accept the graded homework anymore because we need to focus on the final projects. So with that, we want to make sure that, like, from week 1 to week 7, you submit the homework in time. If you miss the homework 1, 2, 3, it's not too bad. It's fine as long as you submit all the homeworks by the week 7 that we can like finalize the grading. And again, if you have any other questions, please feel free to reach out to us in the WhatsApp group and other session, and that should help clarify your question by a lot. And also join our Google group, and also like the regular Google group as well. If you have friends that you are interested in a class, just feel free to share with them as well. I think Bakunda has a question, please.
Finally, I was trying to ask, is this course kind of independent of the other courses? Do I need a lot of knowledge from the beginner course and the 2024 course, or I can actually focus and put my whole energy into this course that is ongoing and maybe have like a lot of the knowledge at the end? Like, do I actually need to be so much dependent on the beginner? Okay, yes, we can go through the videos and understand a bit, but like, how to be too hectic doing 2 things at the same time. So I'm asking, is it possible that you only put your energy into this intermediate course and you gain at the end of the day? Yeah, thank you very much. It's my final question.
Yeah, that's a very good question, Bakunda. I would say that if you understand what I was explaining in this optional session and it just makes sense to you, I think you don't need to go to the first 2 classes to understand everything. But if some of the presentation is not completely clear for you, and you feel like you're gonna benefit more from going back to some of the sessions, I think that would be a good chance for you to go into some of the recordings. I wouldn't recommend to go through all the recordings and all the presentations of those like 6 or 7 weeks before the class next week, because that's gonna be a lot, right? I would say if there is any part that you're interested in, go back and take a look at those, just so that you're in a better position to understand what is going to be provided in the next class. We are trying our best to make sure that, like, even though you don't take the first 2 classes, you can still stay engaged and work together with us in the core class. But again, it's going to be very hard to not rely on any other existing materials. So there might be some reference to the previous lectures, and if there are, we will try our best to suggest you which recordings or which class that you can look at, so that you can look at only like the slide of that class, or like only a tiny bit of that class to understand more, and come back to learn on the core class that is going on like live synchronous right now. I hope that helps.
Yes, thank you very much.
Also, to add on something, all our communications are from our official pages on LinkedIn, on Twitter, and on our email. Please, I request you don't be dragged in any other, like maybe WhatsApp community group. If you need any information regarding our WhatsApp, please email us and we shall give you that information. Don't just join any random group which is not from us. I request, because we don't delegate anyone to work on our behalf. We use our direct communication channels. Thank you.
Yeah, so if you have like a small group because you want to form like a special project, like different from the ongoing class, I think that's not an issue at all. So like, we love that you guys work together and do something. But if you are going to focus on the class and you need some question, you need some clarification from our end, it's a lot better to be part of the key group. Oh, there's a very good question coming from Oguranti. So the question is, how are we going to know those that will be accepted into this course? So we are trying to accept everyone as long as your submission to the registration form is valid. So if you have submitted and everything works for the Google invitation, so you should get an email to show you that you are invited to join the Google group already. So if you haven't seen it, you may want to check whether you have it or not. And if you don't have it, please connect with us. Amrita, do you have a question? If not, I'll keep talking. But if you have a question, please feel free to unmute again. So I'm gonna go back to this slide real quick. All right. Let's see. I hope it works here. So I'm gonna share this slide of this page. This is the one that I have issue with. So, you know, this email from Google should be sent to you, should be sent to email that you use for the registration, asking you if you want to be part of the SynBio4ALL Plant 2025 group. If you accept, then you're good to go. And just please join us on the first class and the ongoing class, and then you're good to go. If you haven't seen this email, contact us or make sure that the email that you use for the registration is working fine. We may add the newcomers, the people who just registered recently today, to the group later, maybe by the end of the week. But if you have any issue, please reach out to us. And we're gonna keep the registration form open maybe until the start of the class, the latest, the start of next week by the latest. After that, we're probably gonna close out the form just to make sure that we can organize and be mindful of who are we, how many people are in our class, because that's gonna affect the size of our class significantly. Yeah.
Okay.
So yeah, go ahead, Kato.
Also, another thing which I would like to echo about is about the registration. If you would like to keep getting our information regarding our webinars, please, you can also tap there and register to get our information. Also, to remind you that on Friday, on 6th of June, we shall be hosting Professor Katalin, who will be talking about much more about plant synthetic biology, because he's an expert. So please, we are going to send out the registration form very soon. I think earliest by tomorrow, or by tomorrow. And if you're interested in listening on what he's going to be talking about, please, you can register for it. Yeah, thank you.
Yeah, so we actually have other webinar going on, and you can take a look at that on our website as well. Let me try to go to that page and share to you right now where it is. And while I'm looking for the webinar page, I want to make sure that you're, in terms of the recording, is going to be available in the syllabus document. So you will be able to see the syllabus document on our website. But we will change that into the public one later. So you would go into the syllabus. The recording link is going to be put right here. Oops, sorry. I hope you can see my slide, right? You can see my my browser right now. But all the slides and the lecture recording and the homework will be available right here. Keep in mind that we're gonna change, this from the website to the public one soon, just to make sure that the information that we updated is still local and manageable for now. but you have access to all these syllabus within the Google group. And this is where we have our monthly webinar. So last year we have several section going on. So this is like the talk that we have when we kickstart our SynBio4ALL system. In 2024, we have like a lot of invited speakers to talk about, you know, some kind of like carbon capture. Like we have people from the company, people from the university to come and share with us what is going on. So it's a diverse group of invited speakers. They are all SynBio stakeholders. So if you're interested, please, feel free to take a look at what is going on. Recently, Sebastian Kuchoba joined us in the webinar last month, and that went pretty well. So he has like a very interesting discussion. Sebastian worked at his house to work on synthetic biology. So if you're interested, please check it out. All these recordings are publicly available in YouTube. And the other webinars is going to be available on YouTube as well. So check it out. All right. I think that's all I want to share. And again, if you want to stay, on top of these, information with our community, please make sure that you join this like general Google group as well. This is like by your choice, if you want to join, just click join, or we're going to send out a form to those who attend this class and also like be part of our class for the registration form for the webinar as well. So there'll be a link in that webinar that asks you whether you want to be part of this Google group. So if you say yes to that one, I will invite you into this community as well. All right, I think that's all. I just want to make sure that we are aware of the time. I think we are doing relatively okay by focusing on the class for just 1 hour and take the rest of the time to discuss on the question and other things. So if there is nothing else, then we will just keep this presentation as is. And if you have any other question, please feel free to reach out to us on email or via our active WhatsApp group. So I think that's it. I'm gonna stop my screen sharing right now. So since we have a pretty good number of people here, maybe it might be a good chance to take a maybe kind of group photo together. If any of you were able to turn on your camera, I think like we may have a new group photo to put onto our website. You can keep your camera on, and I think Kato can help.
Yeah, if you can turn on your video, please. I will look for the one like I will snap out all the nice ones, and maybe we shall look for one to put on the website. Maybe be part of this history. Just turn on your video, and I will snap out the best one. I'm just going to count from 5, and then I will make a snap. Okay, I'm starting to count. 5, 4, 3, 2, 1, and then I snap. Okay, maybe I can take another one. Maybe you. Okay, let me first save this. Okay, I take another one. 3, 2, 1. And then, okay, thank you so much.
Thanks a lot, everyone. It's nice meeting you all. See you next class.
Thank you.
Thank you.
Okay, thank you very much. I'll see you next week in the next class, and also hope to see you for the webinar on 6th. Keep engaged. Please send us email at symbio4all@gmail.com. If you need to join our WhatsApp group, please don't hesitate to reach out. If you want more information regarding SynBio4ALL Africa, don't hesitate to reach out to us. And finally, this course is proudly sponsored by Experiment Foundation. with our individual colleagues and individual partners who love to see Africa growing each day. And we send our appreciation to everyone who has supported us in all aspects. Thank you so much. See you next week. Please don't miss. More exciting things are coming along the way. Have a great evening. Don't hesitate to contact us. Thank you.
All right. Thanks a lot, everyone. Thanks a lot for showing up. I'll stop the recording now. Bye.
Bye.